Welcome back to the deep dive. Today, we're going to completely flip the script on something you probably think you understand. The immune system.
Right.
When you hear immune system, you immediately think defense, right? Soldiers fighting off the flu, white blood cells, inflammation.
It's the classic battlefield metaphor. We've all used it for like a century.
Exactly. But what if I told you that before you were even born, when you were just a developing cluster of cells, your immune system wasn't acting like a soldier at all?
It was acting as an architect.
It's a huge pivot in perspective.
It really is. We're diving into a brand new paper from Nature Neuroscience, January 2026. The title is, well, it's a mouthful.
Go on, hit me with it.
Spatial transcriptomics of the developing mouse brain immune landscape reveals effects of maternal immune activation and microbiome depletion.
I do love a good academic mouthful, but let's, translate that. We're talking about the origins of neurodevelopmental disorders, things like autism spectrum disorder. And specifically, we're diving into a mystery that has puzzled scientists for decades. Why are these conditions so much more common in males than in females?
That is the core question. The statistics are, roughly 4-to-1, male to female, for autism diagnoses.
4-to-1. It's a huge difference.
It is. And this paper uses some incredible technology to peel back the layers of the fetal brain in mice, specifically to show us how the mother's environment can physically rewire the baby's brain. And like you said, turns out the immune system is the construction crew.
So our mission today is to rigorously unpack this detective story. Sentence by concept. We've got two suspects, a maternal infection and a disrupted gut microbiome.
We have a victim, the male fetal brain.
And we have a weapon, a specific immune signal. But before we get to the crime, we need to understand the tool they use to solve it. It's called MERFISH.
Right, MERFISH, which stands for Multiplex Aerorobust Fluorescence in Situ Hybridization.
Okay, let's stop right there. That sounds like alphabet soup. How is this actually different from, standard genetic sequencing?
Good question.
Usually we just grind up a tissue sample and see what genes pop out. Why wasn't that good enough here?
Well, think of standard sequencing like a blender. You take a piece of the brain, you throw it in the blender, and you get a smoothie.
Okay.
You can analyze that smoothie and say, okay, there's strawberry, there's banana, and some kale in here. You know the ingredients, the genes, and you know how much of each you have.
Yeah, you've lost all context.
All of it. You don't know if the strawberry was next to the banana or if the kale was, way over on the other side.
And in the brain, location is everything. A neuron in the cortex does a totally different job than one in the hippocampus, even if they're made of similar stuff.
Precisely. Structure is function. MERFISH is it's like taking a high-resolution photo of a fruit salad instead of blending it.
Ah, I see. So you can see the strawberry sitting right on top of the banana.
You can map exactly where every gene is turning on and off within the actual tissue structure, down to the single cell. It's incredible.
So they took this high-res photo tech and looked at mouse brains at two specific times, E14 and E18. Why those days?
They're critical checkpoints. E14 is mid-gestation. Think of it as the heavy construction phase. The brain is just churning out new cells in these little factories, the ventricular zones.
And E18?
E18 is late gestation. That's more about placement. The cells have been born, and now they're migrating out to form the intricate layers of the cortex.
And they weren't looking for just any genes, right? They focused on about 500 genes, mostly ligands and receptors.
Yes, and this is crucial. Ligands and receptors are the language of cells. A ligand is the message like a text. The receptor is the phone that receives it.
If you don't have the phone, you don't get the text.
You don't get the message. They wanted to see who was talking to whom.
And here's the first big surprise for me in this paper. Even in a perfectly healthy, normal mouse brain, these immune molecules were everywhere.
They were. Cytokines, chemokines, molecules we usually associate with, fighting an infection. But in the healthy brain, they act like air traffic controllers.
So they're not fighting a war.
No, they're telling young neurons, okay, go left here, then stop, and then build a synapse right there. They're guidance cues.
They even found they could predict where autism-associated genes would show up even before a disorder could manifest.
Yes, genes like GRIN2B, ANC3, these are heavy hitters in autism genetics. And the researchers saw them appearing in very specific spatial layers, all orchestrated by these immune signals.
Okay, let's pause. First recap. We need to get this baseline set before we break the system.
Agreed. So, recap number 1. First, the tool, MERFISH. It lets us see gene expression in space. It's the fruit salad, not the smoothie.
Got it.
Second, the timeline. We're looking at the switch from making cells at E14 to placing cells at E18.
Creation versus placement.
And third, the baseline. Immune molecules are not just for sickness. They are the blueprints and the traffic controllers for normal brain wiring.
Okay, so listener, imagine a construction site where the workers use walkie-talkies to coordinate where to put the bricks. Those walkie-talkies are the immune signals. Now, let's see what happens when someone jams the frequency. This brings us to the perturbations. They messed with the moms in two ways.
Right, two different environmental stressors. First one is MIA, maternal immune activation.
MIA.
They injected the pregnant mice with something called poly-IC. It's basically a fake virus. It looks like double-stranded RNA to the body.
So it tricks the mother's body into thinking it has the flu.
Exactly. She mounts a full immune response, but there's no actual live virus to infect the fetus. It just isolates the effect of the mom's immune response itself.
Clever. And the second stressor?
The second is MMD, maternal microbiome depletion. They gave the mothers a cocktail of antibiotics to wipe out their gut bacteria.
So one group has a fake flu, the other has a wiped out gut. What happened to the babies?
And this is where the results just get so striking. When these babies grew up, they showed distinct behavioral deficits.
Like what?
They were antisocial. They just ignore other mice.
Yeah.
And they showed high anxiety, avoiding the center of the room. But here's the kicker. This happened almost exclusively in the male offspring.
Wait, really? The females were fine.
Largely resilient. The females from the same mothers, exposed to the exact same stressors, showed almost no behavioral changes.
That is wild. The environment was identical, the stressor was identical, but the outcome was completely determined by sex.
Completely.
So what did the brains of these affected male mice actually look like?
Physically, they were distorted. Using that MERFISH mapping, they saw that the deep layers of the cortex, specifically layers marked by a gene called CTIP2, were too thick.
So the foundation was poured too thick.
Exactly. And at the same time, those factories we mentioned, the ventricular zones, they had shut down too early. They saw fewer cells with Ki-67, which is a marker for cells that are actively dividing.
So the brain started making new cells too soon, and the cells it did make got piled up in the wrong layers.
It's like it matured too fast and incorrectly, like pouring a concrete foundation and then immediately trying to put the roof on before the walls are even finished.
And again, this was stark in the males, but not in the females.
Absent to the females.
Okay, so we need to understand how, this is the mechanism part, and it gets a little dense, so stick with us here. How does a fake virus in the mom translate to stop building cells in the male fetal brain?
This brings us to the molecular smoking gun. The researchers found a specific signaling network that was just completely broken in the males.
Which one?
The CXCL12 and CXCR7 pathway.
Okay, let's unpack that. CXCL12, CXCR7, who's the sender and who's the receiver?
CXCL12 is the ligand. It's the chemokine. In our construction analogy, this is a foreman shouting, "Move over there." It's a migration signal.
Got it. So it tells cells where to go.
Usually, yeah. It talks to a receptor called CXCR4, which tells the cell to move.
Yeah.
But that's where CXCR7 comes in.
So what does CXCR7 do?
CXCR7 is fascinating. It's encoded by a gene called AcR3, and it's what we call a scavenger receptor, or decoy.
A decoy.
Its job is to bind to the signal, to CXCL12, and basically eat it. It degrades it without sending a signal of its own. It's a regulator. A brake.
Oh, so it's like a sponge. It soaks up the extra signal so things don't get too loud.
A sponge is a perfect analogy.
Yeah.
It soaks up the extra CXCL12 so the cells don't get overwhelmed. Now, here's the finding.
Okay.
In the males exposed to MIA, the levels of AcR3, that's the gene for the scavenger sponge, they skyrocketed.
They made too many sponges?
Way too many, specifically in the young neural precursors. And because they were overexpressing AC3, they were just scavenging all the CXCL12 signal. They were dampening their own guidance cues.
So the foreman is shouting instructions, but the male brain basically put on noise-canceling headphones.
There, it deployed a vacuum cleaner that sucked up the sound waves before they even reached the ears.
That's a great analogy. The signal gets lost. And because those navigation signals are disrupted, the cells don't migrate correctly, and the layers build up wrong.
And you get that thickened cortex we talked about.
And the females?
In the females, this pathway was stable. They didn't spike their AcR3 levels, they heard the foreman clearly, and they built the brain correctly.
Okay, time for recap number 2. Let's lock this mechanism in.
Right. So here are the bullet points. 1, the stressors. We hit the system with maternal infection, MIA, and microbiome depletion, MMD.
2 systems, 1 outcome.
2, the result. Males are severely affected, behaviorally and physically. Females are resilient.
And 3.
3, the mechanism. It's the CXCL12, CXCR7 pathway. The males overproduce the scavenger receptor, the sponge, which is encoded by ACOR3, and that jams their own navigation signals.
Which leads to the obvious next question. Why? Why on earth would nature design the male brain to be so sensitive to this specific pathway?
Hold that thought. The answer is coming up in the genetics section.
But first, we need to talk about social distancing at a cellular level.
Right. This is one of the coolest parts of this study, visually. Right. Because they used MERFISH, they didn't just count the cells, they measured the physical distance between them.
The paper calls it spatial chaos. What exactly moved where?
The most alarming thing involved microglia. These are the brain's resident immune cells. Think of them as the security guards. In the affected males, the microglia moved abnormally close to the migratory excitatory neurons.
So the security guards were crowding the workers.
Uncomfortably close.
Right.
I mean, imagine you're trying to get to your desk to do your job, and a security guard is just standing nose to nose with you, blocking your path.
It disrupts the whole flow.
It does. And they only saw the specific crowding in the males.
And in the microbiome depletion group, was it the same thing?
Similar, but slightly different. In the antibiotic group, certain interneurons were too far away from their factories. So the whole spatial logic of the brain was broken. Things that should be close were far, and things that should be distant were crowding.
Now, a skeptic might say, well, maybe it's just something in the mother's blood that affects males differently. But they proved it wasn't just the mom, right? They took the cells out of the brain entirely.
Yes, this was a crucial control experiment. They took neural precursor cells from male and female fetuses and grew them in a petri dish. No mother, no body, no immune system. Just the cells.
And then they added the signal.
Then they added CXCL12, that guidance cue we've been talking about.
Oh, what happened?
The male cells reacted instantly. They rapidly differentiated into neurons, matured way too fast.
And the female cells?
The female cells largely ignored it. They just stayed as precursors.
Wow. So the male cells themselves are biologically primed to overreact to this signal. It's hard-coded into them.
It's intrinsic. It's not just about the mother's response. It's about how the male DNA is set up to interpret that response. Which brings us to the ultimate question.
Why?
Why? What is physically different about the male DNA that makes it so jumpy?
This is section 5, the smoking gun epigenetics, and this is the part of the paper that really blew my mind. They used a technique called scRNA-seq.
Single nucleus assay for transposase accessible chromatin.
Okay. Another intense acronym. But the concept is actually pretty simple if you think about an open book.
That's a perfect way to put it. Think of your DNA as a library of instruction manuals, but most of those books are taped shut. If the book is closed, the cell can't read the instructions. That's closed chromatin. But if the book is lying open on the table, the cell can easily read that gene and make the protein. That's open chromatin.
So they looked at the DNA of male and female brains really early on E12 before any of the chaos started.
Exactly. And they looked specifically at the gene ACOR3, our culprit, the sponge receptor.
And what did they find?
In the females, the chromatin around ACOR3 was mostly closed. The book was shut.
And in the males?
The book was wide open. The chromatin was accessible.
So in a normal situation, it doesn't really matter. But when the stress hits, when the mom gets that fake flu, the male brain is just sitting there with the instruction manual already open to the page that says panic response.
That's it exactly. It is epigenetic priming. Because the DNA is already accessible, as soon as that stress signal arrives from the mother, the male machinery can instantly start cranking out ACOR3.
But the females have to, what? Go find the book, cut the tape, open it to the right page.
Right, which takes time and energy, so they effectively dampen the whole response. The males react immediately and excessively.
And that overreaction crashes the entire system.
It creates a domino effect. Male DNA is open at ACOR3. Mom gets sick. The male brain cranks out way too much ACOR3. The navigation signal, CXCL12, gets scavenged and confused. Neurons migrate incorrectly. The cortex builds up too thick. And eventually,
you get the behavioral deficits we associate with neurodevelopmental disorders. It connects everything: the gut, the immune system, the DNA, and the brain structure. It is a complete chain of custody.
It really is. And it highlights that sex in biology isn't just about hormones like testosterone. This is happening at the level of how DNA is packaged in the nucleus, long before puberty.
Let's bring this all together in our final synthesis.
Okay, so to summarize, this paper used a combination of MERFISH for spatial mapping and epigenetic sequencing. They found that the maternal environment, an infection, a lack of gut bacteria, directly regulates how the fetal brain gets wired.
But it does so through immune signals. And males are way more vulnerable because of this epigenetic priming. Their DNA is just physically more open at the ACR3 gene, which makes them hypersensitive to these immune cues.
And this gives us a molecular explanation for why autism and other NDDs are 4 times more common in males. It's not social, it's not just diagnostic bias. There's a fundamental biological susceptibility rooted in how the male genome is packaged.
So why should you, the listener, care? Beyond the amazing science, what does this change?
Well, first, it solidly links the gut to the brain through the immune system. It confirms that a healthy maternal microbiome is critical for fetal brain development. Wiping out the microbiome has similar effects to a viral infection.
That has huge public health implications for antibiotic use during pregnancy.
Huge. And second, it raises this provocative evolutionary question. If this priming is so dangerous for males, why hasn't evolution fixed it? Why keep the book open?
That's the million-dollar question.
It is. Maybe. Maybe the sensitivity is a feature, not a bug. Perhaps having this open chromatin allows males to adapt faster to certain environmental changes. In a high-stress world, maybe you want a brain that builds itself differently, faster reaction times, different wiring.
But in the context of modern stressors, it tips over into pathology.
It becomes a high-risk, high-reward strategy.
It's like a sports car with a hair-trigger gas pedal. Great for racing, terrible for parallel parking.
Exactly. And the female strategy is the reliable sedan. It gets there safely, no matter what the road conditions are like.
Before we go, I want to throw a question out to you, the listener. If we know the mechanism is this specific ACR3 receptor, could we theoretically intervene?
I mean, it's a tantalizing possibility, isn't it? If you could design a drug that temporarily blocks ACR3 or somehow closes that chromatin book just during a critical window in an at-risk pregnancy, could you prevent these changes?
We're not there yet, but for the first time, we have a specific, tangible target. And I highly encourage you to check the show notes. We've posted the heat maps from the paper. You have to see these social distancing maps of the microglia.
Oh, they're incredible.
It is chilling to see them crowding the neurons in the male brains. It really brings the data to life.
It does.
You've just taken the deep dive into the neuroimmune landscape. Until next time, keep your chromatin open.
But maybe not too open.
And keep your curiosity high. Thanks for listening.